Okay, mini lecture 2 for this week of November 16th to 20th. So this is what I want to talk to you about in this mini lecture, is the sort of the technical basis for, I understand now why this doesn't do that. Okay, the technical basis. First thing is to think about way back to our early lectures about the production function, so the combination of inputs that produce agricultural outputs. So we think about the land on which food is produced, the labor that goes into it, including the management of that labor and the whole enterprise, capital like our tractors and no-till equipment, fertilizer, and we talked about 4 different types of, we did think about inorganic or organic fertilizer. And then things we're going to focus on today are about genetics. So the genetics which is within the seed which is planted, insect control, and weed control will give special emphasis. So just a few words about science and technology and fertilizer. I guess just to start with recognizing that growing plants need nutrients, especially nitrogen, potassium, and phosphorus. Some of those nutrients are in the soil, and depending on the origin and type of the soil, some of them may be depleted over time through continuous cultivation, and they may need to be replaced. They can be replaced through organic sources like nitrogen-fixing plants, like legumes, and trees like my Phaederbia albida that I've introduced. It can also, nitrogen especially can, we can get it through decaying plant material, which has itself kind of gathered up nutrients from soil, or manure from livestock. So those are organic sources that we can have. The other ones are the main type, really, which fuels our food production, or inorganic sources of nutrients, like potassium that we get from mining potash. And province of Saskatchewan is very, very rich, deep deposits of potassium, which are mined and then made into potassium fertilizer. Phosphorus, which is also has a mineral source, and the largest deposits of phosphate rock are in Morocco and Algeria, and in Africa, Syria, and the Middle East. China has smaller deposits, about 5% of the world's total, but it's actually the largest producer of phosphorus fertilizer in the world. Great strides were made when the world kind of discovered ways to extract, get nitrogen from the air by nitrogen fertilizer from the air in the form of ammonia. So NH3 is ammonia, and the Haber-Bosch process, which was discovered in 1913, converts nitrogen from the air into ammonia through a reaction of the N2 plus H2 using a metal catalyst. With high pressures and high temperatures, you can have 3 units of H2, a unit of N2 produces 2 units of ammonia. This requires plentiful and cheap energy for this process because of the high temperatures and heat required. And Alberta is a very good location for that because of our plentiful and cheap energy sources such as natural gas. So Alberta has a very large, is a very large producer of ammonia fertilizer. Okay, so that's what I'd just like to say to end this off, is that these are relatively uncontroversial technologies. We know that plants need these nutrients, and these are relatively accepted that we can bring these these mineral deposits like potash and phosphorus, mine them from these phosphate rocks and from deep deposits of potash, combine them into fertilizers, inorganic fertilizers for our crops, and that we really depend upon the nitrogen from this Haber-Bosch process that produces ammonia fertilizer. We see from the global planetary boundaries that we don't have new sources of nitrogen. We're drawing down nitrogen, but we have, so therefore we're in a sense over a planetary boundary. We really have a fixed supply of nitrogen, but it's a very plentiful supply of nitrogen relative perhaps to the supplies of cheap energy that we have. So again, this is relatively uncontroversial. We know that we need it. What's more controversial is the amounts which are used and the runoff of these fertilizers into water bodies. Okay, so therefore we're going to focus on technologies which are more controversial. And first, I'm going to talk about plant breeding and what's controversial about plant breeding. And I'm going to acknowledge Dean Spanner for several of these slides and material on these slides. Dean talks about an 11,000 year history of plant breeding by artificial selection. So that's if we think about the population of, say, palm trees, which are on the left. If we want to have palm trees which are bigger and grow higher, then we might select those two which are on the right-hand side of that, have them breed together to produce a new population. That new population will still be variable, but it will have more of the traits of these taller varieties. So over time, from generation to generation, if we keep selecting the tallest two trees out of each population, they will keep producing taller and taller trees. They'll also produce some shorter trees. We keep selecting for the taller and taller trees, the overall population will become taller and taller. And that's what we think of as artificial selection, is on the basis of quantitative genetics and plant breeding. It's fairly simple in a way. And we can see some of the great achievements of traditional plant breeding with the example of maize. It had a wild ancestor, which was, we could find in Mexico. Over years, Aztecs and Incas domesticated that to corn as a staple crop. It was adapted to conditions in Europe, and now there's continual genetic improvement, getting corn which is higher and higher and higher in yield. So that's been done through breeding practices which have been around for thousands and thousands of years. Something which is a bit shorter is the idea of developing hybrids. And so this is sort of displayed in this diagram. Think about the two parent lines. So in the previous slide, I had two kind of high producing parent lines. Here we think about two parent lines which have desirable characteristics for different reasons. And we inbreed them so that they are well known to us of what they are. And then we choose parent lines which have some contrasting characteristics, breed them together to create a hybrid. There is something when that happens with two inbred parent lines, is the first generation of crop plants which are produced from that, called hybrids. They exhibit something called hybrid vigor. That is, they're kind of healthier and stronger in those characteristics than in the parent lines, and they tend to be uniform. So each F1 cross tends to be the same as another F1 cross. Ironically, if you cross the two F1s, you get a whole variety of results in the second generation, the F2 generation. So that means if you want to have crops that have the F1 characteristics, then you need to keep going back to the parent lines P1 and P2, crossing them, and then producing the hybrids. If you try to save the seed and plant the seed, then you're going to get a whole variety of outcomes from the F2, and most farmers would like to have nice uniform crops and that they can count on. So therefore, farmers are willing to pay more for hybrid varieties because of the vigor and because of the uniformity of the outputs that they produce. So we think about that use of hybrid vigor is something that was a part of developing of new varieties of the dwarf varieties of rice and wheat. We've before talked several times about Norman Borlaug as seen as the father of the Green Revolution, who developed these new dwarf varieties of rice and wheat. And when those were combined with irrigation and inorganic fertilizer, as well as appropriate extension advice to farmers, led to year-on-year increases from 2 to 4% for years and years from the 1960s through the 1980s, 1990s. And he was awarded the Nobel Prize for that. And we also talked about the Borlaug hypothesis, and that's named after Norman Borlaug. We have this hybrid varieties, a particular type of breeding. So not all breeding is hybrid, but it has that characteristic. Hybrid varieties tend to be the varieties which are sold by private companies because they can keep farmers coming back to them. Open pollinated varieties which are not hybrid tend to be sold by public companies because farmers can use the seed year after year and plant the seed out. There's been a historical focus, as we talked in the case of Norman Borlaug, on increasing yield, that is output per unit area. Since the 1990s, there's also been emphasis on breeding in disease, insect resistance into the crops, and resistance to herbicides. So that has come since about the early 1990s. Since about the year 2000 or so, there's been more concern about breeding for drought resistance. It's been fairly difficult to do, but some successes have been made. And from about the same time, about the year 2000, there's been breeding for improved micronutrient content. So things like higher iron, higher vitamin A, which are the main type of breeding, which is about changing the taste or texture or consumption attributes of the food, where everything else is really about production. Consumers may benefit from production, innovations in production, but mostly from lower prices relative, and producers often seen as kind of the main beneficiaries. So that's conventional breeding, including hybrid breeding for hybrids. Two other types of breeding which are more controversial. One is transgenic, and this is probably the most controversial. That's when we take genetic material or DNA from one species and we insert it in the genetic material of another species. So that's often called genetic modification, or in a sense, it's a particular type of genetic modification, which is transgenics or between two species. There's the diagram on the right, which helps us understand that, where we have the yellow gene confers a characteristic that is desired. If we have a parent material that has the desired gene and we want to bring it into another variety which has other desirable characteristics, like that commercial variety with the open dots, we breed them together and we get a new variety which has some of the characteristics of both parents, but hopefully including that very desirable characteristic, which is that shown by the yellow dot. So that's a traditional plant breeding approach. The transgenic approach would be to actually identify the specific gene, this yellow dot, not just not among a whole set of DNA, but a particular gene as a yellow dot, and then insert it into a commercial variety so that the commercial variety has all of the original characteristics that the commercial variety had, plus that desirable characteristic which is conferred by that gene shown by the yellow dot. So we're actually literally moving genetic material from one species of organism to another. Genetic editing or gene editing is a different type of genetic modification, which is catching on more and more all of the time. This doesn't involve moving genes between species. It mostly is about kind of turning on and turning off and cutting and pasting material in a particular genetic material in a particular species. So this shows, a little diagram shows kind of snipping and moving out an undesirable gene. They might also want to put in a desire or do something which turns on a particular gene. So that's what we would call genetic editing. And there's a technology called CRISPR, which is often used for gene editing. Most people, very few people actually understand the difference between gene editing and transgenics. Those who do tend to find less reason to object to gene editing because it's really just it's not mucking about with cross gene species, but more turning things on and off within a gene. So that's a little story about kind of the technical basis of breeding. I want to talk about the technical basis of pesticides. And so here are 4 groups of pesticides. So these are all chemicals, or in some cases, biological organisms, which are manufactured into some type of solvent, or they're a produced chemical that are used. Pesticides is kind of the broadest term. We use to refer to chemicals that are applied to protect plants from weeds or fungi or insects. Herbicides are used to control unwanted plants in a field. Insecticides are used to kill insects that might cause damage to a crop. And then fungicides are used to kill fungi. Fungicides are very, very important in Canada here because of the fungus, which are problems with canola and pulses. So field peas and lentils and chickpeas are particularly bothered by fungi. We do quite a lot of agriculture here is focused on the use of fungicides. So that's think about pesticides. The other issue is weed control. So here is a sort of history of how we've controlled weeds. So if weeds are growing in a field that you're also trying to grow a crop in, you get a lower quality crop when you harvest. You also have the weeds which may compete for all of the nutrients and sunlight and water with a regular crop. How do we control weeds? Up until the 1800s and early 1900s, mostly we were doing it by tillage. So let the weeds grow in a field, till them up so that the plant dies and withers in the sun. Do that for a year, then plant your crop. The weeds will slowly come back as the seed is brought in, the seed regerminates or is brought in from outside. So you may have to till or cultivate the field once every 2 years or once every 3 years. And that's what we did when I was a kid, was mostly tillage. Around World War II, there was quite a bit of chemical experimentation to develop both insecticides and herbicides. The herbicide that was developed was 2,4-D for controlling broadleaf weeds, and it was widely used up until the late 1960s. During that time, from World War II until the late 1960s, over 100 different chemical compounds were developed as herbicides, which was a huge investment. And during that time, there's a large increase in use of 2,4-D. Since the 1970s, the Monsanto company investigated and developed a chemical called glyphosate. which was sold under the brand name of Roundup and still sold under the brand name of Roundup and other names from other companies, since the patent is now expired. And we'll talk about sort of the whole story about glyphosate, its success and its challenges. So the history of use of insect control, so that was about herbicides. Now I think about insecticides. There was the first development of a major insecticide that would sort of kill insects. This was DDT. The experimentation into DDT was done to protect troops, thinking about American troops were fighting wars in tropical countries especially, and ways to try to kill the insect vectors of diseases like malaria and typhus. And that led to the discovery of the insecticidal action of DDT. 23 years later, Rachel Carson wrote Silent Spring, which was a book which raised concerns everywhere about the environmental effects of DDT. And after 10 years, it was banned in the United States and is banned in most countries now, but not all. During that 1960s period, when it was looking like there would be seeing these negative effects of DDT, then companies started looking for alternatives like natural pyrethroids. These are insecticides which are extracted from chrysanthemum flowers, which is a type of lily, and then development of synthetic equivalents to the pyrethroids. So these are the new kind of a new generation of less harmful insecticides compared to DDT. The more recently has been the development of a class of insecticides which are neural active, so they act on the neural pathway of the insect, called neonicotinoidal insecticides, or neonics. And these act similar to nicotine, which is why they have the name that they have. Starting about 10 years ago or so, there has come to be increased concern about, sorry, I have to remove this, about the accumulation of neonics in water bodies. So their environmental effects, because they are persistent and they are accumulating in water bodies like the prairie potholes that we have here, as well as even greater concern about their possible role in colony collapse disorder and effects on bees. And the science seems to indicate that neonics kind of cause the bee to be drunk and maybe not be able to find its way back to the colony. So they leave in the morning, go and feed, and they're unable to kind of return to the colony at the end of the day, and the colonies collapse as a result of that. This is lots of debate still about the need to ban neonicotinoidal insecticides. They've been banned since about 2013 in most of Europe. They've been banned in Ontario and Quebec since about 2018, 2019. They're still available here, but there's still some discussion about that, about whether they should be allowed because of their environmental effects. So I'm just going to just to kind of pause this technical session to think about, okay, what we saw. So what have I described? We have described the technical side of this, but I've also described the role of the private sector. So much of this development of plant breeding, as I said, hybrid varieties can be developed by private companies because they can own the parent lines, and farmers need to keep coming back if they want the advantages of the hybrid, the F1 cross, which exhibits this hybrid vigor and exhibits homogeneity or similarity of each crop type. We also see that certainly the insecticides and the herbicides are things which can be, private companies can develop profits from. So therefore they will be quite active in research for those. Other types of innovations which don't provide such clear options for private companies to benefit will need to be developed by the public sector. And so this is one of the challenges, is how much is the public sector willing to invest in food and agriculture innovations, and where should that investment be? And when possible, should there be an attempt to try and draw in the private sector? And we can see it a little bit in the case of the development, the co-development between the government and the big pharmaceutical companies to develop the new vaccines, that it really harnessing that innovation from the private sector has been fundamentally important for developing the new vaccine candidates that have been announced in the last 2 weeks. One of the attractions for the private companies with saying things like the new chemicals, or say glyphosate, is the ability to obtain a patent. So that's a sole right to exclude others from making, using, or selling an invention that holds for a limited time. So Monsanto, after they discovered glyphosate, had I think it was a 12-year period during which time they had the exclusive right to produce and sell glyphosate. These can be really effective when individual buyers, so buyers of the product, so if it's farmers or others, receive all of the benefits. So some of the benefits don't kind of spill over into other people in society. They also work well when it's easy to exclude non-buyers from benefits. So if I don't need to buy your product to benefit from your innovation, then you're unlikely to innovate for me. If the buyer needs to buy new material each production period, that provides a steady flow of income to the company, who will keep then investing and producing that product for years into the future. And when there's a pretty short period between investment and return. So in new innovation, which requires long time lags between investment and return, will be less attractive to the private sector. And in all of those cases, then we would need public investment in innovation. And the issue is how can that public investment is limited by sort of the realities of taxpayers in particular period. So how can that be best managed? So I'm going to take a break now and then come back with the third mini lecture.